Oh, so we might wait for Jim Day and Paul. Paul said he might be 5 too. But Estee, we can use the chat to talk if if we need to. The audience can't read the Zoom chat and they can see whoever's camera's on.
Okay.
So if you turn your camera off, you disappear from the live feed. So I'll ask 3 speakers to give just a really short summary of their talk or any updates they'd like to give first. And that's probably easiest if just one speaker has their camera on, and then we can all just listen. And then when we have the discussion, if you all turn your cameras back on, you can have all 6 of you together.
Does it matter which background we choose? And how do you get it into the, so I've got the PowerPoint.
And then you pick speaker and then add your details.
So how did you do yours, Estee, then? The white one?
Yeah, so you should get a PowerPoint presentation with about 4 or 5 backgrounds.
Yeah, my presentation doesn't have that background that you've got.
I think it's because I'm a respondent and not a speaker.
Oh, I didn't.
Choose one in 4. Enter your name, affiliation, and Twitter handle.
Estee, how did you manage to get yours normal size? Sorry, I realise this is not important.
It was just, it came with I just edited the PowerPoint presentation and I deleted the ones that weren't mine, saved it as a PNG, and then uploaded it as a background.
Well, mine's upside down now. Okay.
Oh, this is so difficult, isn't it? Just for me anyway, because I'm really hopeless at this sort of thing.
Me too.
Very good at genome sequencing, so you know.
Oh my goodness.
Hi, Jim Day.
Oh, yeah, good. Just trying to get this sort of
Hello.
thing kind of, yeah.
Hi, Jim, how are you?
Hi, I'm fine, and you?
I'm very well. You're looking very smart.
Oh, wow. Thank you very much. Nice meeting the other panelist.
We're just trying to get our background slides sorted out, and it's very, very stressful. Very stressful because
I gave up. I tried. It makes my my image sort of like a zombie type of thing. So.
Oh, we don't want you being a zombie. No, but I kind of like a weird landing on Earth person with lights going through their head. I'm going to try and change that in a minute.
Oh, you've got fancy backgrounds, Kate. Wow, you've got super fancy backgrounds.
I don't know what I did, but
have you got background jealousy? I stay there. You got background jealousy.
I just used the one I was sent.
I didn't know. My link didn't work. That's why I've ended up with separate ones, I think.
Oh, let's have a look.
The kind of thing it was doing to me, it looks like it does to ST as well.
Yeah.
I know what I could do.
So once you get the I've got it in the PowerPoint, I've edited it. How do you go and get it into?
To save it as a picture.
Exactly.
This is
and then
PNG or JPEG.
That looks even worse. My goodness me! Look at that.
It has to be a JPEG. I tried TIFF. TIFF didn't work, so it have to be a JPEG or something.
JPEG or PNG.
Yeah, exactly.
And you save the actual slide itself. Did you delete everything else?
Yes.
Yeah, I deleted the other slides.
Oh, I've managed to get my names the right way around, but I'm still.
Oh yeah, you have.
Yeah. Once I tried, it was flip-flopped, so the thing was upside down. So I tried a number of ways of trying to get this stuff to work, but well, maybe I should try one more time.
I think. Yeah. Golly, this is quite stressful, isn't it? I wish I'd done this beforehand. And how did you get understanding dynamics ecosystem? Where did that come from?
Well, that's an unknown.
What?
It was on the template that I was sent.
Yeah.
Wow.
No worries. I think we're going to run out of time in a minute, Sharon, so I will cut your losses. I'm doing this. So I can't find the way to set up your background. I've got the image now. So,
Okay.
So it's on your video settings, there's a little arrow next to your video,
oh, yeah,
and
I've
it's
got that.
video settings,
Oh,
and
yeah.
then you go down to background and you can add it.
So how does it look?
Oh yeah,
Oh, perfect.
that's good. Yeah.
Good.
It's fine?
Yeah, it's looking good.
Okay.
So you saved it as a JPEG.
Yes, I saved
Can
it
you do it-
JPEG.
Can you do it as a PDF?
I think it has to be JPEG or PNG.
Yes.
Oh,
So
Nick.
otherwise, when you try to
PMG. I
upload
haven't
it-
got PMG. When it says save, it doesn't say
Oh.
PMG.
Nick.
We got it.
Oh, green.
Hi, Paul. Now Paul's like, what have you all done?
Yeah, can I have the background, please, Alison?
It's in the chat. If you open the chat.
We are truly stressed trying to do this.
Yeah, I know.
Don't worry if you can't.
Well, when I was doing it by myself, it was not working well. So if you think this picture is fine, then I'll keep it.
Yeah, looks good.
You look
Okay.
fine. I think
Where
I
is the chat?
Kate, yours is backwards, Kate.
I've got these like jokey things. No one else has these silly tags that they made you put in on the thing.
yours is a mirror image.
Yeah, I've been there as well. I've
Okay.
been there as well. I can tell you.
I'm just
Yeah.
going to reach a state of zenness, and I'm going to give up on that.
I'm still trying really hard to get mine uploaded. How do I upload it? Up positive. Okay, I might even be there.
I can't see the earlier
Add
chats,
image.
Alison, I don't think. Can you
Okay.
repost it?
Editing skills.
Oh yes.
In the
Oh,
Zoom.
let's get rid of the lights. Look at this.
Alison, did everyone have this figured out yesterday?
That's better. That's better. I think, yeah, maybe my background is not. Anyway, maybe my background is not showing it. I think I look like an alien, so I'm not going to worry more than I normally do.
I'm pleased I managed to do that. I didn't find how to save it. I just put a screen grab, put it on my desktop.
Yeah.
Have I got any typos? Oh, flip. I haven't put consortium, have I? Oh, golly. I'm going to do that
How are
again.
you, Paul?
I think I'm okay. I don't really know. I think it sounds like all this discussion now sort of just describes the inside of my mind at the moment, actually, which is lots of things going on at once, coming together at the last moment. I can't find the JPEG, Alison. I don't get the early chat.
Oh, okay.
Nothing before I logged on is there.
Um,
I have to repost it. How the hell do we do that?
I can do that. I've got it here.
We are just so proficient. It's fantastic,
You have
isn't
3
it?
minutes.
Ah, brilliant. Thank you.
good luck.
There we go. My full title. There.
Yay.
All right, so we're nearing the time to go live. So I'll ask you all to turn off your videos and mics, and I'll play this little video. And then at 2 o'clock, as soon as the video is over, feel free to turn on your cameras and your mics and begin your panel discussion. Good luck.
Thank you.
Hello everyone and welcome to our session on genomic surveillance. It's a topic that has been put in the spotlight over the last year during the COVID pandemic. It's in the headlines with the discovery of new variants and their implications. But genomic surveillance is not just important for COVID. Pathogens that cause malaria, cholera and a whole range of other diseases need to be and are being monitored to study their evolution, track their spread and any emerging drug resistance. So I'd like to firstly introduce our speakers and our three respondents. We have Professor Sharon Peacock, who's Director of COG UK, Director of Science at Public Health England, and Professor of Public Health and Microbiology at the University of Cambridge. Sharon is going to be speaking about the genomic surveillance of the SARS-CoV-2 virus in the UK. We have Professor Nick Thompson, who's Group Leader at the Wellcome Sanger Institute and Professor of Bacterial Genomics and Evolution at the London School of Hygiene and Tropical Medicine. Nick will be speaking about the global genomic surveillance of bacterium that causes cholera. And we have Dr. Abdoulaye Jumde, Associate Professor of Microbiology and Immunology at the University of Bamako in Mali. Jumde is going to be covering genomic surveillance of Plasmodium parasites that cause malaria. And our three respondents are Professor Paul Kellam from Imperial College London and Kymab Limited, Dr. Kate Baker from the University of Liverpool, Dr. Estee Turuk from the Bill and Melinda Gates Foundation. If you have any questions during the session, please use the box on the webpage just beneath the video. So first, we're going to start with hearing from each speaker to give a short summary of their talk and any updates on the work. And we'll start with Professor Sharon Peacock. So I'll hand over to you, Sharon.
Thank you very much. Well, I don't think I've ever been in a situation before where I'm going to give an update on a talk that's so out of date so quickly. It's really quite extraordinary because the field of COVID-19 pathogen genomics it's moved so quickly since I last wrote the talk that I'm going to quickly go over the contents of the talk and then actually give an update on where we are at the moment. So my talk was really about cracking the code and conduct of the SARS-CoV-2 virus. And in my talk, I described the consortium that we've built in the United Kingdom. So that was first developed back in April 2020, and it consists of a large network of sequencing capabilities now, 16 regional labs across the country, and our 4 public health agencies together with the Wellcome Sanger Institute. Now, we've just reached our 250,000 genome milestone, which is something that we're very proud of. In my talk, I talked about some of the ambitions that we have. Really, that was around tracking outbreaks by SARS-CoV-2 and also looking for viral gene mutations. Now, in my talk, I talked about transmission of very different lineages into the United Kingdom. I described how you can use that to really understand the origin of those particular viruses. And in our case, in the first wave, they were from Europe, largely, actually. And then we showed that in the second wave, we cleared our old lineages and a new set were introduced by people coming back from their summer holidays, actually. I also talked about transmission analyses in hospitals. I then went on to describe about mutations from a viral perspective and from a human perspective. Now, I think the key thing for me is to always think about 4 possible things that the virus could do in terms of its changing. And I think of the word tilt, it's just a kind of memory thing. So is the virus changing so it's more transmissible? Is it changing so that it's avoiding our immunity? Is it becoming more lethal? Or is it changing the way our diagnostic tests operate? So I kind of keep those 4 things in my mind. And much of the work that we've done is on the spike protein of the virus. That's the part of the virus that interacts with human cells, attaches to the cells, and it is integral to it being taken up by the cells. Now, in my talk, I talked about some mutations. I talked about the first one that really came to attention. That's D614G in the spike protein. And that was like a proof of principle or a starter, really, to understand how mutations can really change the way a virus actually can behave. This particular virus was more transmissible, and nearly all of the viruses that we see have this particular mutation in spike, so it obviously increases transmissibility. In my talk, I also mentioned a particular lineage that appeared to spread quite rapidly across Europe over the summer. first detected in Spain, but in fact that's likely to have been through travel. But what I'd like to say is that most of that is in the past, and I'm going to spend just 1 minute perhaps talking about what we're doing at the moment. So really what we're focusing on is variants of concern in the United Kingdom and elsewhere. Now, I'm sure you will have heard that we detected a variant in Kent in the south of England back in, it was sort of towards the end of November, beginning of December. And when we look back, it'd been there since the 20th of September. Now, this was a really unusual SARS-CoV-2 virus in that it had 23 different mutations, so it's quite different to the viruses we were seeing around and about. And the key thing that we noticed was that during quite kind of stringent lockdown, there was a huge surge in cases in Kent compared to other parts of the country. And over time, we managed to put together the story that the viral variant appeared to be associated with transmissibility, and evidence was demonstrated to show that that was the case. Now, that variant has spread across the country in the United Kingdom. It causes about 90% of all cases of COVID-19, and it's spreading across the world now, so it's in many different countries. The other variants of concern that we're closely watching are a variant from originally detected in South Africa. We refer to that as B1351, and a variant first detected in Brazil, which we call P1. Now, the importance of that is that not only do they have a mutation associated with transmissibility, but they also have a mutation that's associated with evasion of the immune response. And we've heard a great deal in the news about studies of the vaccine efficacy. We can discuss that later during questions perhaps, but these variants are of concern because of their ability to reduce the impact of immunity. Finally, just to say that our own homegrown variant, the Kent variant, is now busy mutating in that background. And we've now found variants that actually have the gene associated with, the mutation associated with potential immune evasion, the E484K. So to finish, I would say that this is a very fast moving field. I learn something new every day. Every day there's something different about what's happening with the virus. I think what that means is that we're going to be in a cat and mouse game with the virus as we look at our vaccine strategy, tweak our vaccines, think about booster doses in the future. But I remain very optimistic, actually, that with our ingenuity, that vaccines are going to stay ahead of the game and that we will bring this virus under control. So I'll stop there. Thank you.
Thank you, Sharon. I'd like to hand over to Jimdy.
Greetings, ladies and gentlemen. My talk is on how we can use genomics to help with the control and elimination and ultimate eradication of malaria. Malaria is still a major killer in the world, and especially in sub-Saharan Africa, where over 90% of the morbidity and mortality of the disease is still in sub-Saharan Africa. After a few years of steady decline, in the last several years, there have been a stalling of the decline of the disease, and data indicate that the goals set by WHO for 2030, we are pretty much going off track of reaching these goals, at least especially as far as sub-Saharan Africa is concerned. So we need new tools, new ways of doing things, and better ways of doing things. And we think that genomics is such a new opportunity that we need to tap into to not only devise new tools, but increase our understanding of the disease, the pathogenesis, the drug resistance, the transmission, and the spread of the disease. And the recent data indicate that this is indeed can be a game changer. There are observations that were made 10, 20 years ago, where some vaccines that were very efficacious in Latin America were only 30% efficacious in East Africa, and only 3% efficacious in West Africa. We didn't know why this was. Now, thanks to genomics, we know that it's linked to the diversity of the malaria parasite that can lead to this type of differences in response to vaccines and to various interventions. So genomics bring a new stream of knowledge that would allow for a better targeting of the parasite and the disease. And developing countries, especially in sub-Saharan Africa, need to take advantage of this new technological development to apply this new knowledge to solving the problem of malaria. That's what we have been trying to do in our network, the initially called Plasmodium Diversity Network Africa, PDNA, that eventually evolved to become the pathogens, the Genomic Diversity Network Africa, a group of about 16 African geneticists that are coming together and taking advantage of the diversity of malaria in a larger sense, because there is diversity both in transmission, in not only in the genetics of the parasites, but in transmissions, the epidemiology, and in the socio-economical aspects across the continent. All this diversity need to be taken into account to study, to investigate the blueprint of the malaria parasite, so as to come up with new tools and better ways of understanding the spread of the disease, the development and spread of drug resistance, etc. And we are conducting these studies in an unprecedented way by opening up to each other, by bringing together our common know-how and our common set of samples, and sharing information, and leading to discoveries that were not possible when we were each working in silo in our respective places. So we think this is the way forward, working together and taking advantage of each other's strength and opportunities to harness the promise of genomics and genetics, and to bring that to bear on the malaria. In addition to the technical and scientific side, we are also engaging with the policymakers and the National Malaria Control Program officers, and so that they have a better understanding, a better buy-in into the new promise that genomics is bringing to bear on the issue of malaria in sub-Saharan Africa. Over.
Thank you, Tunde. I'd like to hand over now to Nick. Nick, I think, Nick, are you back?
I am. Sorry, I just dropped out there. That's classic Zoom. Okay, so good afternoon, and I thank everyone for attending and the opportunity to speak. So my talk was really about cholera, Vibrio cholerae, the bacterium that causes cholera. And of course, the work that we've been doing resonates very strongly with both Jimdy's work on malaria and, of course, Sharon's work on COVID-19 and the impressive impact that genomics has had in understanding the evolution of that virus just within the 12 months. Cholera, of course, is a historical disease. And so whilst actually, amazingly, there are huge levels of resonance between COVID and cholera. It's really been an enigma in terms of the genomics and the understanding of population structure and what causes cholera in terms of the lineages responsible for epidemic disease within the species. So what we did and what I presented were how we'd used whole genome sequencing to understand the population diversity of Vibrio cholera. And really what we did there was to move away from classical typing strategies that have been used for decades to really understand the epidemiology of cholera and as a proxy for understanding the diversity and spread of Vibrio cholerae. And what we were able to do is move away from those old typing. So, for example, Sera group 01, which is used really to identify, for many years has been used to identify high-risk Vibrio cholerae, to one where we could infer the phylogenetic tree, so the phylogenetic structure of the entire species. And we could use that as a framework, a genetic framework, then to hang the genomes or isolates taken from key events globally that describe the patterns of disease in the last pandemic. So that's the 7th pandemic, which began in the 1960s. And what we were able to do is to show which lineages were connected to those historical events. So, for example, the large-scale epidemics in Latin America in the 90s. Or the first set of cases in Africa in the 70s within pandemic 7. And we were able to connect them using that phylogenetic tree to say how closely related they are, or they were. And what we were trying to do was to understand really how populations as disparate as perhaps the west side of Latin America related to populations of cholera-causing disease on perhaps the east side of Africa. And what we showed was actually that not all lineages within that, not all branches or lineages within that phylogenetic tree contributed equally to disease. And actually, in truth, some lineages were much more important. And actually, during pandemic 7, a single lineage accounted or described or even explained much of the epidemic disease we'd seen, epidemic and pandemic disease we'd seen within pandemic 7. And what that did was actually call into question some of our previous dogma, really. It built on those dogma, and it called into question our idea that these were local environmental organisms that sort of moved. There was an ingress from the environment into the human population during outbreaks, and that was all that explained large-scale outbreaks and intercontinental transmission, to one where clearly, if a single lineage had spread within 60 years, multiple times globally, this was pretty rapid, and it isn't consistent or wasn't consistent with this being a localized endemic problem as such, and but more easily explained by rapid transmission and carriage, and probably more likely human-to-human transmission or human contaminated environment to human transmission. So what this actually then has done is not only to redefine perhaps where to look within the species, so assign risk to particular lineages, and Sharon spoke about COVID-19 and various different lineages like B117 in the UK. We were able to assign risk to specific cholera lineages, and by doing that, we could then start to, one, redefine definitions of Vibrio cholera, so put Vibrio cholera into cholera, the disease, And we've been able to look at more recently to perform detailed studies where we look at the contribution in the single country of different lineages towards disease alongside the epidemic lineages to understand their relative contribution and potentially their differences. And like Jimdy, we've been working with the Global Task Force for Cholera Control to look at questions such as specifically defining the pathogen you're actually trying to control, to think about regional spread, not just local spread, think about intercontinental and long-range transmission and the role of human carriage in the actual spread of cholera. So simple information like what lineages are responsible for epidemics means that you can actually start to piece together much more complex control plans associated with the control of what is an enigmatic and historical disease that still claims a huge number of cases and lives annually. And I'll finish there.
Thank you. Thank you to all of our three speakers for fascinating talks. Can I invite the whole panel now, speakers and respondents, to join us? And I'd like to invite you to discuss the wider themes of genomic surveillance and delve into a little bit of the detail with each speaker. Paul, would you like to kick off the discussion for us?
I'll try, and thank you very much, Alison, and thank you to the speakers. It's fascinating to hear all that you've had to say. I have a question for Sharon to start with. I mean, what you've achieved in such a short space of time in the UK is nothing short of amazing. The large-scale sequencing, the regional sequencing, the computational biology behind, and the organizational infrastructure. It's clear other countries are doing that as well, and you can note South Africa and Denmark and others that are really applying genome sequencing. But if you reflect on your last year, are there things that you would advise others that have worked really well, things that you would change? How would you do it again?
Well, I've been reflecting on lessons learned, actually, Paul, fairly recently. And some things I think I could recommend strongly in terms of what worked for us. I think that working together as a team, collaboration across the country is what made it work. I think working in silos certainly wouldn't have worked. Sharing everything, so it's a giveaway culture actually, and that can be quite unusual, giving away your data without even analysing it, so you just put it straight out there. We've also given protocols and other things to people. I think that one of the things that I perhaps learned is that that pace over perfection is quite an uncomfortable place to be. I think many of my scientific colleagues are perfectionists. And I think that one of the things we have found quite difficult is that you often have to do things at pace. So if you're going to develop a network across the country and spend £20 million doing it and generate 250,000 genomes, you might think you take a year to plan that. Actually, it took a day for the blueprint, 2 weeks to get the funding, and then we were off. And I think many people who are involved in the response will actually, it will ring a kind of a note of familiarity with them, perhaps, that one of the key things I've learned is that you have to be willing to work at pace and take quite big leaps of faith into doing things that you have to have sort of faith in what you're doing, and that's not so easy. So, and you're constantly retrofitting what you're doing to what everyone else is doing, because you're part of a completely networked system where you have diagnostic labs which feed into sequencing labs. So we service 90 different NHS laboratories plus all of the major lighthouse labs. So I think there are so many lessons, actually. One day I'm going to write a book, Paul, but I think that those two things are perhaps some of the things I'd start to highlight.
Thank you. Perhaps if I can have one more question. So at the moment we are, in the UK at least, hopefully at the top and coming down quite rapidly from the current wave of infection. and I guess the way that genomes will be used as the number of cases start to come down to a more manageable level will change slightly. Could you perhaps give us an insight as to what you're thinking next for the foreseeable future in the UK?
Well, if we could sequence every single genome of every single virus that's causing disease, that would be a special situation. And I would use that information in two ways. I think that you have to bring that data together and collectively say what's circulating in the population, how might vaccines need to be changed in order to protect that population. But then the question is, if you knew that somebody had a particularly worrisome variant, the question is, could you actually do something to protect their family, their friends, and so on? Could you do a really rigorous contact tracing to stop second and third kind of contacts actually acquiring that? So I think it works in two ways, both for individuals, if you can sequence fast enough, and particularly for populations, so that you can get your vaccine strategy right for that population and more importantly, the world, actually.
Thank you very much. So it sounds like there's no rest coming anytime soon.
No.
Thanks, Alison. I'll hand back to you.
Thanks. I'd like to invite our other respondents to question the speakers as well. Esther, did you have a question?
Yes, I do. So, Jimdo, thanks very much for a fantastic talk, as always. I guess thinking about sort of lower middle income settings, what do you see as the opportunities and challenges of malaria genomics, or indeed pathogen genomics, in improving disease control, so malaria control? I think you're on mute, Jimdo.
Sorry. So
opportunities and challenges to use
opportunities and challenges. Yeah, exactly. Opportunities. The field of genomics, as far as it is sort of homegrown in sub-Saharan Africa, is fairly recent. But already we have a number of examples that show that how important this tool is. One example would be tracing drug resistance. Nowadays, quite a number of interventions in the prevention of malaria in children, prevention of malaria in pregnant women, use drugs that for which we no longer do the classical in vivo assays. We type the molecular markers that have been shown to be associated with resistance to these drugs, and that can, in a much faster way, give you an idea of where you can deploy such tools or not, and actually follow the evolution of the parasite in those settings, and to adjust your strategy accordingly. The other low-hanging fruit is in the field of diagnostics. The rapid diagnostics are based on antigens that are coded by genes that have been shown to be deleted in some areas. So if a parasite deletes the gene, even if the parasite is there, the diagnostics now going to see it. So mapping those deletions would clearly give you indication of whether your favorite diagnostic is working or not. So these are really good examples of that are being used. Now there are other examples where we can use the measurement of the evolution of the parasite diversity as a metric of decrease of transmission. And also in R&D, in vaccine development, drug development, we can look at the existing diversity, which gives you information on whether a given vaccine is likely to be efficacious in an area or not. So the genomics is really helping. Now, the challenges are that it's a new technology is fairly expensive. So getting the technology in the hands of people is one challenge. The second is the know-how in terms of analyzing and actually framing the results in a way that is understandable by the disease control community is another challenge we're working on. And getting the appropriate buy-in by the disease community. That is where actually COVID is helping us by breaking down the molecular epidemiology in a simpler way, and that is really helping us in making people to understand what we are talking about when we speak about genetic diversity in malaria as well.
Fantastic. Well, that was a really comprehensive answer, I think. So one of the things is obviously no man is an island, and you don't work in a vacuum. And we're all aware that there are sort of regional initiatives that are developing for genomics in Africa. So how can we capitalize on these, the regional initiatives such as the Africa CDC Pathogen Genomics Initiative, or indeed your own Pathogen Genomic Diversity Network?
Yeah, I mean, Sharon has said it, you need to work together. To be effective and efficacious, we really need to work together. And I think initiatives that, any initiative that bring people together is a must. And Africa CDC being endorsed by the African Union is a very good umbrella under which it's easier to reach out to the entire continent and then synergize and take advantage of the strength and the opportunities that are here and there, so that we can, as a continent, address these issues that any single individual country cannot do on its own.
Fantastic. So I think I'll now hand over to Kate Baker, who's going to interrogate Nick, I think.
Thanks, Esther, as always. Yeah, I mean, thanks to all the speakers for wonderful overviews. It's lovely to see the synergy between them. And I wanted to kind of draw together the theme of thinking about, Sharon, what has been achieved with COVID genomics in the past year is, I feel like the field's been building towards this for quite a long time, and it has completely shown the worth of the process of whole genome sequencing for epidemiology. But I wanted to come back to cholera, Nick, and this is something that we've been chipping away at for a long time. You mentioned it as a traditional diarrheal disease, and so how do you see the future of our work on these older pathogens that we've been dealing with for a much longer time, now that we've had this insanely amazing case study of the use of it in COVID-19? How will it change things?
Well, I think that's great. I mean, it's a great question. And actually, one thing I want to make clear that old doesn't mean unimportant. In many regards, old diseases that still linger and yet are actually distinctly addressable either through vaccination or through use of genomics become, COVID should raise them up the table and actually show what you can do in such a short period of time. And I think that's the main thing it's done. COVID-19 has focused on SARS-CoV-2. It's actually highlighted how important it is to understand the evolutionary biology of the actual infectious agent. And for cholera, we've been dogged a little bit because actually we call it cholera. Not Vibrio cholerae, not Vibrio cholerae pandemic 7 lineage, or any precise definition. It's actually the definitions, and it's actually our understanding of that organism that I feel has really hindered us in many ways. What COVID's done is show that once you actually understand, and the more data you have, the better. Once you understand how it's evolving, you could create your own opportunities to intervene. And it might be that it's to do with having to rethink vaccine formulation strategies, or it may be you can institute different control strategies. Instead of blanket strategies that cover the whole country, you might be able to target that in a more precise way. And I think for cholera, that's what we've been lacking. We've been lacking that deep understanding of the genetics of the organism and how it's evolving. We've been missing opportunities to actually target the pathogen that causes disease in a precise way, and genomics has allowed us to do that. And then I think it also, and again, as the speakers, both Jim D and Sharon have said, it also then allows you to rationally design control programs and then test the efficacy of those. And we haven't been able to do that before. It just disappeared, and we consider it to be a local endemic population that would come back in the next rainy season. The genomics has said that's not true. It actually circulates, it enters countries, it moves regionally and intercontinentally. And I think if we understand the patterns of spread, as we now do for COVID, for cholera, then we'll be in a similar position to COVID. Perhaps after many decades of time working on cholera, will be a similar position that we are for COVID, and hopefully in not too distant future. And I think considering that COVID and cholera evolve at different rates, then the opportunities for cholera control are huge.
Yeah. Well, and I guess on that note, one thing I having gone over your talk again before this, I thought about how is the variation, and both Sharon and Jim Day touched on this in their talks, the variation, applicable to therapies. What is the picture for cholera drug resistance and potentially future vaccination strategies that is shaped? How is this population shaped by those forces?
I mean, the key with all of this is you knowing your target. Actually, one of the cornerstones of epidemiology is to have precise definitions. And so, we now know that really for epidemics, we're looking for a single lineage. Based on that, we can say that actually the O1 antigen, which was used to type cholera, is relatively invariant, but invariant in that lineage, right? So we could just say in general, it's the actual genes that encode the O1 operon are just invariant, but actually they're present across the species. And so it only matters that they're invariant in that lineage. And so that's the first thing. We also know if you look across the species, you can see drug resistance markers across the whole species. But again, within that lineage that causes epidemics, that's really the only place that it's important it's great to have the context, and biologically, and of course as a scientist, I want to know all of this across the whole species. I want to know how the whole thing's evolving. But in terms of therapy, it's only important to know what that lineage has got and what the long-term trends are for that lineage. And we know that actually multi-drug resistance is a feature of cholera, and yet to some extent, oral rehydration and replacing salts are the key treatments to cholera. But we also know we live in a world that's contaminated by antimicrobials, both in the environment and in our gut, if we don't regulate the usage and we use them unwisely. And so cholera, the lineage that causes epidemics, is living within that human environment. And therefore it's being impacted upon by that human environment, which includes antimicrobials. And so we know that actually within the last 2 waves of the current pandemic, global radiations of the 7th pandemic lineage, multi-drug resistance was the main redefining feature of that lineage. And we know that actually in refugee camps in Kenya, for example, we know that cholera became resistant to frontline and last line antibiotics, even though they're not used to treat directly cholera, or at least they're not recommended for treatment for cholera. So these things are impacting on cholera, and understanding how that's transmitted through understanding the actual routes of transmission, the pathways for transmission of cholera, can only help inform strategies for usage, strategic usage of antimicrobials for treating cholera.
Thanks.
A question perhaps for all people on all of the speakers, because you all have different areas that excite you and your domain knowledge about your pathogens of interest. One of the things that was incredible with the UK variant, the B117, was how the genetics and the epidemiology and the modeling alone was able to suggest very strongly that this was a virus that had increased transmissibility without any need at that stage to do phenotypic proof of that. And actually the ability of phenotypes and traditional, if you like, microbiology to keep up with the pace and the data of genetics is completely out of kilter. How do you think this will play out in the future? And is there what is the role for phenotypic biology in this world of genetics with pathogens?
So perhaps I'll go first, Paul. I think that we were always going to need phenotypic information, and I think it's a sort of another strand of evidence. I think that we're seeing we can get very early evidence for example, vaccine efficacy. It's quite difficult to know what kind of a surrogate of effectiveness of a vaccine is. So if you have a result in a neutralization assay, what does that actually mean to a clinical trial? But I think it gives you a heads up, and I think that's important. The other thing is that from all of the phenotypic work and computational biology work that people have been doing, certainly in COG UK and I'm sure elsewhere, we've actually got a list of changes, mutations that lead to an amino acid replacement, which we're watching very carefully, because we know that in phenotypic assays, for example, using pseudovirus, we know that there's some particular mutations that are going to be particularly problematic in all probability if they arise in humans. So I think that gives us a kind of list that we're looking at really carefully, comparing that with our population. And then if they do appear, we've got quite a difficult challenge of deciding whether or not it's going to be important. And so I think there's a fine line between watching what then happens to individuals and doing very early investigations and then acting. I think one of the things that's really surprised me about the pandemic, and perhaps it shouldn't have surprised me, is that we're having to use evidence that's not peer-reviewed. And I think that the challenge for us thinking about the phenotypic assays is actually saying, what does a phenotypic assay in one lab mean to another lab, and are they comparable, and are they using the same control? So I think that I would always want to have phenotypic data. In the COVID-19 response has been really critical, but you also have to be very mindful of the fact that it's quite difficult to compare results from one lab to another. But I think it is making a big difference to us, and it will continue to do so. So we're going to have to keep the labs open, which is, I think, a good thing.
Yeah, maybe I would jump on that, and I also think that yeah, the genetics is important, the modeling is important, but especially in the case of malaria, where we have a parasite that is fairly complex. So the translation of the what you see in the gene into what really transpires in people is much more difficult to establish. So there has been a lot of progress made in this field, but the complexity of the interactions, especially the host pathogen interaction issues, and the redundancy that you can find in our parasite, where sometimes genetics may indicate something, parasite may have other ways of doing the same thing. So measuring the phenotype and actually taking into account the epidemiology is going to still be important. Yet the genetics do give us a heads up, and there is the clear example of the issue of drug resistance, where traditionally we only understand the genetics when people are already dying of resistant parasites in the clinic, while in that was in the old drugs. With the new drugs, we've been able to understand the genesis of resistance through genetic studies, and actually zoom in the Mekong area and pretty much eliminate the parasite, the falciparum altogether, nearly eliminated there. And that certainly contributed to sort of stopping the spread and the spread of drug resistance, which traditionally comes from that area to Africa. And so although the genetics is greatly improving, we'll still need the labs to better understand what is going on in reality.
I believe the recording's about to end, Nick, so keep going and we'll see where we get to.
No worries, thank you. And I'll give you one very quick, I'm not sure how long this will go on for, but I'll give you a very different example. We know with cholera, pandemic 7 is called by lineage called 7th pet, but the previous pandemics 1 to 5 are caused by a separate lineage, a discrete lineage called classical. We know that actually that phylogenetic, they're closely related, but if we look in the lab, actually they're phenotypically, there's considerable differences. There are key differences between them. So what we know is that cholera, pandemic cholera has emerged twice. Now, whilst we can follow the